a channel dedicated for science and critical thinking, hands-on skill and compassion, ethics and teamwork, lifelong learning and innovation. Learning starts now with me and you.
Hello everyone, welcome to our virtual class. I'm Tony, and today we're diving into a topic that happens after DNA has been transcribed into RNA, the post-transcriptional modifications of RNA. But before we jump in, let's quickly go over what we'll achieve by the end of this lesson. First, you will be able to define post-transcriptional modifications as essential steps in mature RNA formation. Second, you'll be able to identify the three major post-transcriptional modifications: the 5' capping, the 3' polyadenylation, and RNA splicing. You should be able to recall and list these three specific modifications by name, and if presented with a description or diagram, correctly name them. And finally, you will be able to state the primary consequences of defective RNA processing. So without further ado, let's get started. Have you ever wondered what happens to the genetic information after it's been copied from DNA? It's not as simple as just making a copy and sending it off. Imagine you're building a complex machine. You wouldn't just take the raw materials and expect them to work perfectly, right? You need to refine them, shape them, and assemble them precisely. The same goes for RNA. So you might be able to recall from our previous lessons that transcription is the process where a DNA template is used to synthesize RNA. You might think, great, we have RNA, let's make some proteins. But actually, there's a whole lot of action that needs to happen first. So let's kick things off with a quick question to get your brains warmed up. What is the primary product of transcription from a DNA template? Take a moment to think about it. If you said RNA, specifically pre-mRNA, you're absolutely right. This pre-mRNA is like a raw, unedited draft. It contains all the genetic information, but it's not yet ready to be used. This is where post-transcriptional modifications come in. They're essential steps that transform this raw transcription into a mature, functional RNA molecule. These modifications are vital for the RNA stability, export from the nucleus, and ultimately its translation into proteins. Let's start with the first two major modifications that act like protective gear for our RNA molecule: the 5' capping and the 3' polyadenylation. Imagine our pre-mRNA as a delicate manuscript. If we just leave it unprotected, it's vulnerable to damage, right? That's where the two processes come in. First, let's look at a simple diagram of a pre-mRNA strand. Now I want you to think, what happens after transcription? Can mRNA be translated immediately? Well, the answer is no. The initial RNA, which we call pre-mRNA, cannot function properly unless it's modified. To further understand why, let's first begin with our first modification, the 5' capping. This happens at the very beginning, or the 5' end, of our RNA molecule. A molecule called the 7-methylguanosine cap is added to this 5' end. So what's added to the 5' end of pre-mRNA, and why? This cap serves several crucial purposes. It protects the RNA from enzymes that could break it down. Without the cap, the RNA would be quickly degraded. Next, it helps ribosomes recognize the mRNA so they know where to start the protein building process, and it assists in moving the mRNA out of the nucleus and into the cytoplasm where the protein synthesis occurs. Now think about this. What do you think would happen if the 5' cap is missing? Without that protective cap, mRNA would likely be destroyed before it even has a chance to be translated, leading to a host of problems in the cell. Next, let's transition to the 3' polyadenylation process. This is like adding a long protective tail to the other end of our RNA molecule. A long tail of adenine bases, sometimes hundreds of them, is added to the 3' end of the RNA. This is called the poly-A tail. We call this process 3' polyadenylation, and the resulting tail is known as the poly-A tail. So what do you think is the function of the poly-A tail at the end of the 3' end of the mRNA? Similar to the 5' cap, the poly-A tail has vital roles. It also protects the mRNA from degradation, it helps the mRNA move from the nucleus to the cytoplasm, and it assists the ribosome in knowing where to start translation by affecting the initiation of protein synthesis. Interestingly, there's a specific instruction on the pre-mRNA itself that signals where this tail should be added. This instruction comes in the form of a signal sequence called AAUAAA. So what does this signal do during polyadenylation? Think of it as a molecular stop sign or a flag that precisely makes the spot for the enzymatic machinery to begin adding that long string of adenine nucleotides. It's a crucial recognition sequence, ensuring that the poly-A tail is added at the correct position. Finally, after our pre-mRNA has received its protective 5' cap and its stabilizing 3' poly-A tail, what would we now call this RNA molecule? We'd call it a process RNA, but crucially, it's still often referred to as a pre-mRNA at this stage, especially before the next major modification. It's closer to being mature, but not quite there yet. In summary, the 5' capping and the 3' polyadenylation help protect the RNA and prepare it for translation. They are important for the mRNA stability and its journey from the nucleus to the ribosome. Now, let's move on to arguably the most complex post-transcriptional modification, RNA splicing. You might be asking, what happens to pre-mRNA before it becomes mature mRNA? If you remember, our pre-mRNA transcript contains both coding regions and non-coding regions. The coding regions are called exons, which contain the information that will be translated into protein, and the non-coding regions are called introns, intervening sequences that are not part of the final protein code. So based on that, what do you think is removed during RNA splicing? If you answered introns, that's right. During RNA splicing, the introns are precisely removed and the exons are then joined together in a continuous sequence. This process is absolutely critical because those introns would disrupt the final protein sequence if they weren't removed. This cutting and pasting operation isn't random. It's carried out by a molecular machine called the spliceosome. The spliceosome is a large and complex molecular machine composed of small nuclear ribonucleoproteins and other proteins as well. Here's how the spliceosome works. First is the splice site recognition. The spliceosomes first recognize specific consensus sequences at the boundaries of each intron. These are like molecular signposts. The most common signals are a GU at the 5' end of the intron and an AG at the 3' end. There's also a branch point A site within the intron that's crucial for the process. Second is cutting. Once these sites are recognized, the spliceosome orchestrates two sequential cutting reactions. First, the 5' end of the intron is cut, and this end then loops back and attaches to the branch point A, forming a lasso-like structure called a lariat. Next, the exon joining. The 3' end of the intron is cut, releasing the lariat. Simultaneously, the two neighboring exons are ligated or joined together with extreme precision. This ensures that the genetic code remains in the correct reading frame. Now, a reflective question for you. Why is it important to remove introns? If introns remained in the mRNA, they would lead to the production of non-functional or even harmful proteins. But wait, there's more. RNA splicing isn't just about removing introns. It also allows for something called alternative splicing. Imagine you have a set of building blocks, and you can arrange them in different combinations to build different structures. That's what alternative splicing does. From a single pre-mRNA transcript, different combinations of exons can be included or excluded, leading to the production of multiple distinct protein isoforms from a single gene. This is a major reason why humans, despite having a relatively modest number of genes, can produce a vast array of proteins and functions. A great real-life example of alternative splicing is the tropomyosin gene, which produces different forms of the tropomyosin protein found in various tissues like muscle cells. Now that we've covered the normal process of RNA processing, which is 5' capping, 3' polyadenylation, and splicing, let's talk about what happens when things go wrong. Defective RNA processing occurs when there are errors in any of these crucial steps. These errors can have severe consequences, often leading to various human diseases. Here are a couple examples of human diseases linked to RNA processing defects. Splicing defects are a common cause of disease. For instance, familial dysautonomia is a rare genetic disorder caused by a splicing error that affects the nervous system. Spinal muscular atrophy, or SMA, a leading genetic cause of infant mortality, is often due to a defect in splicing. Polyadenylation site mutations can also lead to diseases such as certain forms of alpha thalassemia, a blood disorder where the body doesn't make enough of a protein called alpha globin. While less common, cap methylation defects can also impair proper RNA function. These examples highlight just how vital accurate RNA processing is for maintaining our health. When these finely tuned molecular processes go awry, the consequences can be significant. So to recap, remember these key takeaways. Post-transcriptional modifications are essential for turning raw RNA transcripts into functional molecules. Next, 5' capping adds a protective cap and helps ribosomes recognize mRNA. 3' polyadenylation adds a poly A tail protecting mRNA and aiding its nuclear transport and translation. RNA splicing removes introns and joins exons, and alternative splicing allows for the production of multiple proteins from one gene. Defective RNA processing can lead to various diseases, underscoring the importance of these precise molecular steps. Okay, that's all for now. I hope this session has brought you a clearer understanding of post-transcriptional modifications of RNA. Should any questions arise as you review, remember you can always revisit the materials on our YouTube channel or connect with us directly. With that, we conclude today's session. Have a wonderful day and happy learning. Oh, wait, but before you go, what's one new thing you learned today about this lesson? Share it in the comments below. Thank you.